Good afternoon, everyone. Thank you for joining us for yet another session of the SAIMM MTM and this time the GCRF Mind, Dust and Health Network webinar. Just to take us through some of the few courtesy rules. If you're not comfortable with your name appearing because this meeting is recorded, may you kindly switch on your name, switch it to anonymous. And we'll have a Q&A session after the presentation. So please, as this webinar is going on, type your questions in the chat forum and we'll attend to them after the presentation. And those that will want to raise live questions, please feel free to do so during that time allocated. You can raise your hand and you can contribute to the discussion. So let's make it interesting. Before I introduce the speaker of today, I'm just going to give us a slight overview of where we are coming from. Could you confirm that you can see my slide? I'll move this so that I can get.
We can see your slide. Yeah, we can see it, Cledwin.
So those that remember the very first forum, this was the introduction that we are a collaborative interdisciplinary think tank. That's what the Mind, Dust and Health Network is about. And just an overview of what we've gone through this year as we are winding up the year. Our first presenters detailed mitigating long-term dust risks from tailing storage facilities. And that was given by Professor Dylan Randall and Helen Marie. We moved on to organizing fence line communities in the context of mining. That was from Brown Matsau. Then jumps to the United States through an escalation of black lung disease in the US and a systematic approach for controlling the respirable diseases that was given to us by Jay. And then we came back to SA with Mr. Sibusiso Masanaho, who gave us the MOSH dust leading practices as critical controls in the South African mining industry. And just recently, we had Professor May Hermanus, who was giving us detailed information around the Chiamiso Trust, where it originated from, the mandate that it was given, the uncertainties, ethical concerns, and food for thought. And as if to wind up the year, we started with tailings facilities. We're now closing off with tailings facilities. So our speaker for today is Dr. Shalene Andraus, who's going to speak to us on hazard identification and exposure of assessment of dust emissions from South African gold mine tailing sites. I will stop sharing at this moment and go back to introducing Shalene. Shalene has a PhD in hematology and molecular medicine from the University of Witwatersrand. She started her career as a medical scientist with NIOH in 2010, specializing in particle toxicity and nanotoxicology. She's registered with the South African Council for Natural Scientific Professions and as a professional natural scientist in the toxicological science domain, and she's also a member of the Toxicology Society of South Africa. Now we are welcoming Dr. Shalene Andraus. Shalene, it's your time.
Perfect, thank you. Thank you for that introduction. Let me just share my screen and please tell me if you can see it. Okay.
We can see it.
Okay, perfect. I think that, should I keep my camera on or off?
What you're comfortable with, Shalene? We can interact with your face as well. It's really nice.
Okay, let's see how that goes. Okay, if we have issues with bandwidth, then obviously I will switch it off.
Okay,
no, thank you. Thanks, Cledwin. Okay, yeah, so as Cledwin has mentioned, the title of my presentation is the Hazard Identification and Exposure Assessment of Dust Emissions from South African Gold Mine Tailing Sites. Okay, so now we all know that gold mine tailings dumps, or also known as tailing storage facilities or TSFs, contribute largely to high dust levels in the Gauteng and Northwest provinces, and are seen as a public health concern for surrounding communities. Now, indeed, human populations residing near these TSFs have raised concerns regarding high dust levels and the onset of respiratory-related symptoms likely caused by tailings emissions. So now the big question is, are surrounding populations at risk for developing adverse health effects due to inhalation of tailings dust emissions? Now, in order for us to attempt to answer this question, we have devised a project consisting of 3 main aims. The first aim is to conduct a hazard identification. So with this aim, we wanted to answer the question, do tailings dust even exhibit an inherent characteristic to be toxic? So for this, we determined the toxicity of tailings dust in vitro. So this is with in vitro cell lines. And we also identified the physicochemical properties of these tailings dust likely to govern toxicity. So then our second aim was to conduct an exposure assessment, because of course, even in our first aim, if we see that yes, the dust show toxicity, exposure assessment is extremely important, because even if they do show toxicity, if the surrounding populations are not exposed to the dust, then obviously there wouldn't be a risk for the surrounding populations. So now with aim number 2, we were trying to answer the following question: are surrounding populations even exposed to tailings dust? Now for this, we looked at 2 different populations. We looked at residential areas close to tailings sites, as well as schools surrounding these mine tailing sites. We looked at 2 different PM levels. So we looked at PM10 levels as well as respirable levels. So in this case, I would just refer to respirable as PM4. We also did real-time monitoring of our PM10 levels, as well as filter collection of our PM4 levels. So now obviously with aim number 1 and aim number 2, what we could then deduce was the risk characterization of the tailings dust. So with this, we wanted to ask answer the question, or ask at least the question, are populations at risk for developing either non-cancer or cancer adverse health effects? So for this specific aim, we assessed the silica levels from the filters collected at each of these schools. And with these levels, we calculated the non-cancer and cancer-related risks using models that we got from the US Environmental Protection Agency. Okay, so these were the 3 main aims of the study. I will also, at the end of this presentation, I will touch on disease prevalence, as this was also done by one of our collaborators that we had for this project. Okay, so now for this project, we looked at 5 TSFs in total. 4 of these were located in the Johannesburg-Gauteng area, and I will refer to them as TSFs 1 to 4. And we also had a 5th TSF, which was located in the Klerksdorp Northwest area. So this is TSF 5. Okay, so now for the remainder of this presentation, I will be discussing each aim separately. I will be discussing the methodologies used as well as the results obtained. So I will be moving from our hazard identification, moving on to exposure assessment, ending with risk characterization, and then touching on the assessment of disease prevalence. Okay, so now hazard identification. So for this, the main idea was to collect the samples, assessing the physicochemical characteristics of the dust, and then also looking at the in vitro toxicity of the dust. So you are more than welcome to have a look at this publication. It was published in Inhalation Toxicology, and this section contains all the details, or this publication contains all the details with regards to hazard identification. Okay, so now, bulk dust sample collection. So the dusts were collected randomly on the top surfaces of the TSFs with a shovel, approximately 10 centimeters below the surface, and this was transported to the lab in 5-liter plastic buckets. So now the figure on the right shows the collection sites as black dots. And then all these samples were received to enrich the airborne particle fraction likely to be inhaled. Okay, so now once the samples were collected and sieved, physicochemical analysis was done on the samples. So first of all, we looked at size distribution. And this was analyzed using a scanning mobility particle sizer, as well as an aerodynamic particle sizer. We looked at the surface area of the dusts. So that was done with the BET nitrogen adsorption. We looked at the shape using scanning electron microscopy. And we also obviously looked at surface elemental composition, and this was done using energy dispersive X-ray spectroscopy. We also looked at the total mineral composition using XRD, and we looked at the total elemental composition using ICP-MS. And then lastly, we wanted to look at the surface activity of the particles, and this was done using an electron spin resonance spectroscopy. Okay, so now the main results in terms of the size. Okay, so what we could see is that sieved dust samples contained particle sizes in the thoracic as well as in the respirable range. So now, if we look at table 2, this basically just shows that tailings particles from all the TSFs have the ability to enter and deposit in the deeper regions of the lungs. And this was assessed using the multiple path particle dosimetry or MPPD model. So this model estimates the percentage deposition of particles if they were to be inhaled. And you can have a look and see in which regions the deposition would be. So in this case, in the left side of this table, this shows the percentage deposition in the tracheobronchial region. And then on the right, it shows the percentage deposition in the gas exchange or alveolar region. So obviously you can see that percentage depositions range from about 3% all the way to about 18, 19%. So that is now whether you look at the median mass aerodynamic diameter or the count mode diameter. Okay, so the point here is that it obviously would deposit into your lungs once inhaled. Okay, so now moving on to the minerals. So clearly several minerals were identified, including the different crystalline silica polymorphs. So this included quartz, tridymite, chrysoberyl. But tridymite and chrysoberyl was quite low, as you can see here. It was below detection level in certain cases. Obviously, quartz was the most abundant, and this ranged from about 70% to 80% in all of our tailing sites. Okay, so then our major surface elemental components, so that expectedly consisted of oxygen and silicon. This obviously made up our silica. We also saw concentrations of aluminium as well as iron. So now this was quite interesting because the dust samples from TSF4 showed quite a high maximum of iron concentration, iron percentages. So this is 67.8% for TS4, and then this was followed by 47.9% for TSF2, which you can see here in table 4. So this was quite interesting, seeing the high concentrations of iron. So then expectedly, we saw arsenic in all these samples. We saw gold. We picked up cadmium. We also noticed or identified nickel and of course uranium. So you're more than welcome to have a look at table 5 in that publication that I showed you, because this shows all the different elements that were identified in these samples. Okay, so now with this slide, this just shows the scanning electron microscope images of a representative set of tailings dust samples. So this is just one sample from each TSF. And I just want to show you or draw your attention to all of these irregular shaped particles. As you can see, especially here where my arrow is pointing, I'm hoping that you can see this. And then also, I mean, this was seen in all the samples. So clearly it is very irregular and it contains sharp points. So you can just imagine the damage that this would do once inhaled. Okay, so now the surface activity. So the surface activity just basically refers to the ability of the particles to produce reactive oxygen species or radicals once inhaled. Okay, so this was then assessed using the ESR. So a higher ESR signal would indicate a higher ability of the dust to produce radicals once it is inhaled. Okay, so now for this, we always use a positive control. Our positive control is Minisil 5. This is a crystalline silica, and this is known to contain a high surface activity. So we would then compare our results for the dust samples with Minisil 5. So as you can see, Minisil 5 has quite a high level of surface activity. And then this is then followed by the dust from TSF4. So you can see that it is comparable to that of Minisil 5. So TSF2 also contained a high level of an ability to produce radicals. And this basically just, again, contributes to toxicity if radicals are produced once inhaled. Okay, so then based on the physicochemical analysis, we then moved on to looking at the in vitro toxicity of the bulk dust. So we used two cell lines. We used a human bronchial epithelial cell line as well as a U937 monocytic macrophage cell line. So now we chose these cell lines because it represents the first level of defense in the human body once particles are inhaled. So we have an epithelial model as well as a macrophage model. So it is a good representation of what you might see in the body. Obviously, we again included our positive control, Minisil 5 crystalline silica, and we assessed the toxicity using an Excelligence real-time cell analyzer. Okay, so I won't go into much detail about the principle of the Excelligence, but the point here is that you have a normalized cell index against time. And an increase in cell index would indicate that your cells are viable, your cells are happy, they are growing, they are doing well. And a decrease in cell index would indicate that your cells are detaching from the wells. So this means that toxicity has been induced, or the cells are just not adhering to the surface. So clearly something is happening. Obviously, you would compare this to your untreated cells. So now for the bronchial cells, the epithelial cell line, you can clearly see that all the dust samples showed toxicity. Obviously, our Minisil 5 showed the highest toxicity, and this is expected as this was our positive control. Looking at the macrophage cell lines, you can see that toxicity wasn't as apparent as it was for the bronchial cell lines. And this is also, in terms of literature, looking at literature, it is known that macrophage cell lines are not as sensitive as epithelial cell lines. So this was also sort of expected. Again, our positive control Minisil 5 showed the highest toxicity. But clearly, there was a bit of toxicity when looking at the macrophage cell line. Okay, so now, based on these results, we could perhaps answer the following question. Do tailings dust exhibit an inherent ability to be toxic? In our case, probably yes, although, again, we were only looking at in vitro assessment. We didn't move on to looking at in vivo assessment, so we didn't, we only treated cell lines, we didn't treat any animal models, we didn't continue with that. But based on these results, we could probably say that yes, probably these dust samples do contain an inherent ability to be toxic. So then we moved on to our second aim, and that is looking at exposure assessment. So this section of the project was also published, and this was published in Clean Soil, Air and Water. And you're also more than welcome to have a look at the detailed information in this publication. But the aim here was to firstly look at PM10 sampling. So for this, we collected ambient filters, and we also collected continuous real-time data measurements. We looked at residential areas surrounding 3 of the 5 TSFs, and we looked at 2 seasons. We looked at the wet season, which is October, and also the dry season, dry windy season, which is August and September. We also collected a total of 22 filters. Okay, so now in this slide, continuous real-time ambient PM10 levels were recorded at 1 minute in intervals using the GRIM environmental dust monitor. And this was located in the backyards of private urban residences. So then this figure on the right, it shows the sampling sites here indicated with the star icons. And as you can see here, the sampling sites were located about 300 meters to as little as 175 meters from the TSF. So then, of course, we also collected weather parameters such as wind speed, wind direction, and rainfall. And this was recorded by climate sensors that were attached to the GRIM monitor. Okay, so now looking at the results of TSF1. So the daily averages of mass concentrations of PM 1 to 10, as well as the daily average wind speeds, were recorded between October 1 and October 31st, 2012 at TSF1. So now the dust levels are indicated in the gray line, and then the wind speeds are shown in the red. So now with this, we compared our results to the current 24-hour national ambient PM10 standard of 75 microgram per cubic meter. Obviously, this level will change. I think it will change in 2030, but for now, this is the current level for PM10. And as you can see here, the levels or this specific standard was exceeded up to 4 times within this 1 month period. And this is shown with the green arrows. So this is the exact same figure, but just with the wind parameters included. So we then looked at the days on which the standard was exceeded. So obviously it was 4 times. And we then looked at the wind direction as well as the wind speed on those specific days. So now for the first dust event, this was on the 7th to 8th of October 2012. And this shows that according to the wind data, 40% of winds higher than 3 meters per second came from a northerly direction. So now, as you can see here in this figure, our sampling site was located south of the TSF. So obviously wind coming from the north, that would be of interest to us because that would show that the TSF could have contributed to the high dust levels. Okay, so then, yeah, as I mentioned here, as the dust monitor was located south, it is likely that the TSF emissions contributed to this specific dust event on the 7th to 8th of October. We then moved on to looking at the second dust event. So this is then the TSF's emissions contributed to this dust event as dust coming from the northerly direction was recorded. And then looking at the third and the fourth dust events, you can clearly see that the peaks are smaller than the first and the second dust event, and this is likely due to precipitation. We recorded rain on these 2 days. So then again, wind blowing from the northerly direction probably indicates that TSFs likely contributed to these peaks. Of course, it is impossible to rule out any other dust sources. We then moved on to TSF2. In this case, the standard of 75 microgram per cubic meter was exceeded 3 times. We again looked at the wind parameters on these 3 days, and clearly you can see that the sampling site was located in a southwest direction from the TSF, So everything from the northeasterly direction would be of interest to us. So that would indicate that the TSF is contributing to the levels. So then I would say in the first case, yes, we do see that there is dust coming from that direction. Again, in this case, on the 30th of September, we could see the exact same thing. But then on the 19th of October, I wouldn't say that this contributed too much to this high dust level. Moving on to TSF3, we could see that the levels were again exceeded 4 times. So again, we had our 4 different dust events. In this case, our sampling site was as close as 175 meters south from the tailing site. So obviously, again, everything coming from, or winds coming from the northerly direction would be of interest to us. I would say this is about what, northwesterly direction. So I would say that this probably influenced or contributed a little bit to the dust events. But then in the other cases, I wouldn't say too much. Okay, so now moving on to the respirable PM4 filter collection at schools. Okay, so in this case, 1 Gillian personal air sampler containing a 25 millimeter MCE filter was attached to each school learner. So the school learners were aged between 12 and 14 years, and they were sampled during the day while they were at school. So then the figure on the right, this shows the different sampling sites, again indicated with the star icons. And then the level of PM4 as mass per filter was analyzed using gravimetric analysis. So for each school, we collected about, I think it was 10 to 15 filters per school. So then the concentration of the PM4 in air, so that is then in microgram per cubic meter, that was calculated based on the sampling time and the flow rate of the samplers. Okay, so then again, looking at TSF1, in this case, we had 3 schools. The first was located 1.83 kilometers away from the school, and the PM4 level was as high as 120 microgram per cubic meter. The second school was located 2.25 kilometers away from the TSF, and the third school was located more than 5 kilometers from the TSF. So you can see that the concentrations of dust were 80 and 25 microgram per cubic meter, respectively. So now again, following the same approach that we did with the residential areas, we looked at the wind speed as well as the wind direction. And in this case, for TSF1, none of the predominant wind speeds were in the direction from the TSF to the schools. So therefore, the TSF could not have been a source contributing to higher PM10 levels at the sites close to the TSF. Okay, so now the majority of the wind at TSF 1A was higher than 3 meters per second. Okay, so this is now indicated by the red line. And this correlates well with a high median PM4 level of 120 microgram per cubic meter. But this was then likely the result of other dust sources other than the TSF. So the TSF definitely did not contribute to this high dust source. Okay, so in each of these 3 days, no rain was experienced, no precipitation was recorded. Okay, so then I'm also showing the results for TSF3. So in this case, again, we sampled 3 different schools around this TSF. We had 2 schools that were quite close to the TSF. It was about 600 and 155 meters from the TSF. And then we had a school that was farther than 6 kilometers from the TSF. So then for TSF3A, so it's this one, more than 60% of the wind was in the direction from the TSF to the school. So you can see in the south-southwesterly direction, this is where the wind was coming from. So you could assume that the dust levels experienced could have been as a result of the TSF. So then, even though wind was more than 3 meters per second, it is likely that the TSF emissions, or less than 3 meters per second, apologies, it is likely that TSF emissions could still have contributed to the PM4 levels as the school was merely 155 meters from this TSF. And it is quite shocking how close the school is from the TSF. For TSF 3B, again, wind was recorded from all directions, including the TSF, as you can see in this wind rose. So clearly, all dust, numerous dust sources could have contributed to this high level of 105 microgram per cubic meter. Okay, so now looking at our results. Are surrounding populations exposed to tailings dust? And I would say probably yes, although other dust sources also likely contributed to the exposure. In our case, it was impossible to separate the TSFs as a dust source from any other dust sources out there. It is quite a difficult task. And the other problem that we also had is that each school could only be sampled on one day. So that means that we only have dust levels from one day. And this was quite a bit of a challenge because we had so many schools. In terms of our residential areas, we at least could have recorded dust levels for as long as 1 month periods. But in this case, it could only be one day. So I wouldn't say that you could clearly say that that is representative of what the school is experiencing over time. That was also one of the challenges that we found with the exposure assessment section. Okay, so now moving on to the risk characterization. So in this case, or this part of the project was also published in the Science of the In Total Environment. So you're also more than welcome to have a look at this risk characterization section in more detail. And in this case, our first step was to first estimate the concentration of particle-bound silica. So we chose silica for our risk characterization because it was the most abundant in all the dust samples. We could have done this risk characterization on metals as well. We just didn't get that far. But for the purposes of this presentation, what we did is we quantified the crystalline silica, so that is in microgram per filter, using the MDHS method 101. Okay, so the concentration of crystalline silica per cubic meter of air was then calculated again based on the sampling time and the flow rate of the samplers. So this is the exact same calculation that we use for the PM4. Okay, so then obviously to allow comparisons between schools, we then took the level of the crystalline silica and converted this to an 8-hour time weighted average. Okay, so now first of all, before we look at non-cancer or cancer adverse health effects, so the first aim is to first look to estimate the exposure concentration of particle-bound silica. Okay, so for this, we used a calculation from the US EPA. And in this case, we used the concentration of silica that we got from our filters. And we estimated the exposure time. We said that our exposure time is 24 hours per day. We also estimated our exposure frequency. I will come back to this now. And we also estimated our exposure duration in years. So we decided to use 70 years because we wanted to look at a long-term exposure in this case. And then we also use the averaging time, which is just, this is from the US EPA itself. So now, for the exposure frequency, we needed to know how many days per year the surrounding populations would be exposed to this silica concentration. Now, we used two scenarios. We used a maximum scenario at first. We looked at, or we anticipated that people would be exposed 165 days a year to the silica level. Our scenario 2 is a bit more representative. In this case, we only looked at the days we only looked at days where exposure would be evident. So that is on windy days. So in this case, we also looked at atmospheric dispersion of dust. So therefore wind speed, wind direction, and rainfall. And based on this, we got a number of days where people would more likely have been exposed, as opposed to just choosing 165 days a year. So once we had our estimated concentration, we then moved on to looking at the non-cancer risk to silica. We again use the equation from the US EPA. So in this case, it is literally just dividing your exposure or your estimated concentration by a reference concentration. Our reference concentration is an estimate of the daily exposure to the human population. This is now including the sensitive subgroups that are likely to be without an appreciable risk for adverse effects during a lifetime. So this usually is already set by international institutions. So in our case, we used a reference level of 3. This was set by America for PM4 crystalline silica. So in this case, we literally just took our estimated exposure concentration, and we divided it by 3. So a hazard quotient of less than 1 would indicate that there is no risk for surrounding populations to develop a non-cancer adverse health effect, but an HQ of above 1 would indicate that there might be a risk. So then, keeping that in mind, what we saw, okay, so each of, if you look at this table 6, we have our different sampling sites at the different TSFs. We have our scenario 1, scenario 2, and in this case, we then calculated our hazard quotient. So this is now for the non-cancer risks. And you can see that all these values are above 1. So that indicates that there might be a risk for these communities to develop an adverse health effect, non-cancer adverse health effect within their lifetime. So we couldn't analyze all the schools because certain schools had a really low concentration of silica. In this case, with scenario 2, obviously our levels would be a bit lower because, as you might recall, scenario 2 uses, or it assumes, a lower exposure frequency. So that is exposure to the number of days are less, but still it is again above 1. Now, it is important to assume that a lot of assumptions are obviously made during this risk characterization. You assume the number of hours that people are exposed. You assume the number of days that people are exposed, but it still would give you a nice indication. Okay, so now moving on to the cancer risk. This, again, we just use the calculation available from the US EPA, or an Environmental Protection Agency. So in this case, you use an inhalational unit risk. This is a set value. This is something that has already been established. And in our case, this IUR, it was calculated from a cohort mortality study of South African gold miners who were exposed to gold mining dust composed of mainly quartz. So we took this IUR level and we again just multiplied it by our exposure concentration. So now the EPA again gives a range of what is acceptable. So any values between 1 million and 10,000, that would be acceptable. Anything out of this range would indicate an issue, especially on this side, because it will obviously indicate a higher probability for cancer risk. Okay, so now going again to our table 6 and looking at our cancer risk, we see values of in the 10,000. So that means 1.6 times 10 to the minus 3. So this already shows you that it falls outside of our acceptable range. So again, we couldn't analyze all the schools. Some of them were not applicable because we couldn't get high enough silica levels, but there were certain schools that we could analyze. And you can see that all of these fall without or outside the acceptable range. Again, looking at scenario 2, all these values are again outside of the acceptable range. Okay, so are populations at risk for developing non-cancer and cancer adverse health effects? Probably yes. Although, again, when looking at the limitations, we only looked at silica. So we didn't look at any of the toxic metals bound to particles. And we also looked at inhalation only. We didn't look at any of the other exposure routes, for example, ingestion or skin absorption. Okay, so now just to touch on this section, the assessment of disease prevalence. So I'm just touching on this section because this was conducted by our collaborators from at UP, the University of Pretoria. So this is Prof. Wee's group. And what they did is they looked at disease prevalence in adolescents as well as in the elderly in these communities surrounding these 5 TSFs. Okay, so in this case, looking at the elderly results, they conducted a multiple logistic regression analysis. So this indicated that living close to mine dumps was significantly associated with asthma, chronic bronchitis, chronic cough, emphysema, pneumonia, and wheeze. Okay, so they did, of course, control for all the confounding factors. So they looked at smoking status. So they looked at a whole bunch of other dust sources. They looked at cooking methods of different cooking styles in these communities. They also looked at the disease prevalence in adolescents, as I've mentioned. And in this case, so they could show that communities in close proximity to mine dumps had an increased likelihood of current wheeze, as well as rhinoconjunctivitis. So there wasn't a positive association with asthma, which was difficult to explain in this case. Again, they did look at other confounding factors, as mentioned in the previous slide. And then now moving to the conclusions. So looking at our first aim, hazard identification, we could say that yes, tailings particles showed an inherent in vitro toxicity based on their physicochemical properties. And something that I forgot to mention, but I did mention in one of the slides that 2 of the sites contained really high iron levels. And these 2 sites, it was TSF2 and TSF4, with the high iron levels, we also saw a really high a relatively high surface activity. And it is known that iron results in the ability to produce ROS in the body. So there was some kind of correlation between the physicochemical properties and toxicity. Looking at exposure assessment, ambient PM10 levels in residential areas close to TSF exceeded safety levels of 75 microgram per cubic meter. So this was for TSF 1, 2, and 3. These were analyzed for a month at each site. And then for the respirable PM4 levels of school learners at various distances from the TSFs was high at certain schools, although it was really difficult to deduce whether those high dust levels were as a result of being so close to these TSFs. Okay, so our risk characterization, this again showed that certain schools showed non-cancer risks, certain schools also showed cancer-related risks. And this was then based on the results that we got from our exposure assessment. Okay, so then the acknowledgements. Funding for this project was provided by the South African Mine Health and Safety Council. I'm also providing the code here in case you want to have a look at their website. And yeah, that brings us to the end of this presentation. Thank you.
Thank you very much, Shalene, for the interesting presentation. Now it's the time for questions and contributions. I can see someone in the attendee. I trust Google will shift everyone from being attendees to panelists so that they can participate. Okay, Harold, you've got a question. So you'll see that you're moving from being attendees to panelists. So once the process is done, Harold, you can.
Okay, thank you.
Good.
Can you hear me
Hi,
now,
Harold.
Cledwin? Hello. Charlene, congratulations on successfully completing your doctorate. Now it was a difficult project at times, and although you haven't indicated I was part of the project in the early stages, that there was this fundamental flaw in our thinking that we could sample the schools on the day that there was a high dust event, so we could measure actual exposure, and that in terms of logistics proved impossible. Getting permissions to go onto school premises is incredibly difficult to start with for one day, let alone for 30 continuous days to be able to capture the events. So I think that your interpretation using the GRIM continuous monitor was a correct way to go. So I think it's a slight conceit on the part of the epidemiologist that you actually want to measure exposure on the body rather than continuous exposure in the environment. And the supposition that we had is that school children are in the area, their residences every day, whereas adults go out to work and are not at their residences during the day when the dust is likely to be blowing. However, I'm a little bit concerned about the way that you've estimated your exposure factors. You took the extreme case of assuming the exposure for 365 days a year, and you have already detailed that the mine dust exposure is closely related to high wind speeds, moderated by rain. So that is really an extreme base case. When I look at your risk results based on your scenario 2, I understand that you applied a filter of windy days plus modified rain days, but the reduction in your risk factors is only about 20 or 30%, which means that you must have assessed close on 200 to 250 days of exposure when there was wind-blown dust. Is that correct?
Yes, so that is a really good question, because one thing that we were wondering about was looking at the wind speed and what would be enough to generate particle aerosolization, basically.
Okay, so Charlene, that is exactly my point,
Yeah.
and I'm a little bit disappointed because that figure is very well known to those of us in the dust monitoring fraternity, that lifting speed is a minimum of 16 kilometers per hour, and it goes up sharply. So there is a threshold. So it's not a continuous function from 0. So the number of days on which there's actually a measurable amount of dust, in other words, to distinguish mine dust from all the other sources, you apply a strict filter of the wind speeds above 3.5 or 4 meters per second, and the number of dusty days then goes from what you've assumed of 250 days to less than 15 to 20 days, and that would absolutely change your scenario 2. So your scenario 1 is unrealistic, but for academic purposes, understand why you did it. But you have now published a result in which you estimate that there's a cancer and a non-cancer risk based on a very basic misunderstanding of the meteorology and dynamics of dust generation, and information which is and was widely known by members of the team. So why I'm saying this with a rather emphasis, we've recently had inquiries from the mining industry in the West Rand, because there have been public concerns. A question that was put to me: are there publications that indicate the health hazard related to mine dust? And I'd have to tell them, yes, here is the basis of a study supported by the Mine Health and Safety Council, recent work by the most reputable institution, but the risks that have been estimated are based on a incorrect assumption. If you look at the work of Maseki et al., who was also part of this larger project, we also adopted a pessimistic scenario of taking the highest measured dust concentration during a period when we had, in fact, experienced a dust storm, and we applied it for 90 days of the windy season. Even that is an overestimate, and we calculated the risk, the identical EPA procedures that you used, but in this case for the heavy metals, arsenic and lead. And we found that there were no inhalation or dermal contact cancer risks, even under the exaggerated scenario of 90 days of exposure at the highest level. Now, we didn't do that for silica, so we'd have to merge the two studies, but there is a divergence in the methods, and I hope that in a subsequent analysis and publication that we can get together and resolve this, because this is a matter of these papers will go into public policy, and the policymakers don't necessarily understand these refinements, or they will, especially under pressure from the environmentalists, they will latch onto the fact that the National Institute for Occupational Health said that there's a cancer risk.
Okay.
So sorry to beat up on you, but I guess that's the purpose of this network forum, where we can robustly discuss these things. Cledwin, sorry for taking so much of the question time. I'll give the floor back to you.
Thanks, Prof. Arneman.
Thank you, Harold. Celine, do you have some responses to that, or it's an offline discussion that can continue?
Okay, no. I fully agree. I fully agree that in our case, with regards to the risk assessment, we looked at the worst case scenario. And from an academic point of view, that is how we look at our risk assessments, and that is looking at the worst case scenario so that we can take a conservative approach. So it is much better to say, okay, well, it's much better to say, okay, let's take a conservative approach, and let's assume this much is being, or the levels of dust is this much, rather than underestimating it and saying, okay, well, now there isn't any risk, there isn't any issue at all, and then later on, a few decades later on, we might see a difference. We might see that yes, people were exposed to a higher level of silica. But I fully understand what Prof. Arneman is saying and where he is basically coming from. But in our case, I think I also mentioned it in my publication where I said these are literally worst case scenarios, because we are trying to look at to follow a more conservative approach. But no, I agree with Prof. Arneman. I mean, it would be nice if we could actually sit and look properly at Joel Maseki's work that he has published, and then try and make sense of this. So yeah, I fully agree with Prof.
Okay. Thank you, Charlene. Thank you, Harold.
Oh, yeah. So just one thing I also want to mention is that in terms of our windy days, I did write it down here. In terms of our windy days, looking at the number of windy days, we did look at any dust or wind speeds higher than 3 meters per second. So I think, Prof, you said it is 3.5 or 4, right?
Yeah, they're about 4.4, somewhere there.
Okay. I would definitely want to recalculate this using those values. Okay, yeah, thank you. Thanks.
Thank you. I saw one hand, but then I can't see the panelists anymore. Murad had a hand up, but I guess he's not in the audience. Are there any other questions? Please put your questions in the chat platform. There was a contribution from Peter Osborne. He said in the 70s and 80s, there was a campaign by COMRO to stabilize the dumps, including planting GM grass. What happened to this approach?
Okay, so this is a good question. From what I've read, planting grass is extremely expensive. It is high to maintain. It would actually be quite nice if Prof. Onaban can also jump in here, because I know that at the end of our project, we try to look at recommendations and suggestions. And from what I've read, planting grass is just too much to maintain, and there aren't any funds to carry this out or to maintain this for years after a mine has been closed.
May I come in, Cledwin?
Okay, yes, Harold.
Okay. The program of vegetation did proceed. It was largely successful until the 1980s, when they developed technology for extracting the residual amount of gold, and we started reprocessing the dumps, and while reprocessing, reclaiming the material, you obviously destroy the vegetation cover. So there was the added challenge that when the dumps had been vegetated, urban development was allowed right up to the toe dams of the dumps, and now you took away the protective layer, and the populations and the businesses were far more exposed. The ultimate solution, which takes a long time, decades, was to remove the dumps, and they've been consolidated. We've recently published a paper, and it was in the proceedings of the Clean Air Conference of 2020, showing how the vegetation of the Brakpan tailings dam, that's a very big final repository just south of Springs, has been successfully vegetated and properly done this time from 2007 onwards, when new management, Ergo, took over. They started vegetating right up to the edge of the current deposition, and the dustfall levels dropped dramatically as you see the vegetation. Yes, it is expensive, but as one of the magnificent seminars that you held about 3 months ago with a colleague from the Council for Mineral Sciences, showing how best practice adoption of mitigation is the attitude. So Ergo's willingly adopting best practice rather than reluctant minimal compliance. And this shows that the vegetation, even in active reclamation projects, can significantly reduce the incidences of dust emanation, even when there's high wind. So that vegetation is effective, but Charlene is correct, it needs to be maintained, and the funds for doing that have to come from current revenues. It's no use this preservation fund, that model that was supposed that the government keeps a private savings account until the mine closes. The rehabilitation must be an ongoing process during the life of mine, not taking part of the mine's revenue and putting it frozen until the mine is about to close, and then thinking that you can apply remediation when all the infrastructure is removed. So as this whole dust forum discussion proceeds, we are understanding that it's a dynamic situation. There are solutions, vegetation is working, but it requires a change to the legislation and regulations that currently in force.
Thank you, Harold. I just have one question, Harold. How much of this vegetation will be selected on the basis that it's native to the area where the TSFs are?
Charlene, sorry to be taking your time.
That's fine. That's perfect.
One of my students from University of Johannesburg did a study of the vegetation on a dump that had been vegetated in the mid-80s, and it had a vegetation survey done shortly after it had been grassed for the first time, and then 30 years later she did a detailed botanical study of the species that survived. This notion, and it was the model that the botanical supervisor was advocating, of functional ecology restoration, that you try and restore to the best possible back to the original vegetation. It's a fanciful dream. The silica sand is sterile, it's acidic, and contains very little nutrients. So it's only very tough plants that can survive on it, native races that have adapted to it. So very little of the vegetation that did survive and flourish on the dumps, or not flourish, but survive, was indigenous. A lot of it was windblown seed from surrounding seeds and weeds, rather than of exotic species, rather than of the indigenous vegetation. So no, sorry, it's not a very good environment. One of the things that has been done, in answer to the first question about what happened to the grassing program, the original dumps had very steep slopes, about 30 degrees, which made them very susceptible to erosion. One of the steps that has been taken, especially around those very big dumps near Diepsloot on the other side of the highway, is to make the slopes more gentle by closing in the toe dams and changing the slope from 30 degrees down to about 20 degrees, so that you've got less or a reduced runoff damage and risk, so the vegetation has a better chance to be stable. Thank you.
Thank you, Harold. Thank you, Charlene. I can see we've run out of time. There's one last contribution. Mariette is asking about the recording. The electronic copy will be available on the GCRF Mine Dust and Health Network YouTube channel, so you can go and visit that YouTube page, and you'll find the links are on the Mine Dust and Health Network website page, minedust.org. I cannot see any further questions, and because we've run out of time, I think we will then conclude this meeting. Charlene, thank you very much for coming on board and for presenting such important information and sharing your insights to what you have found in your work. Thank you, everyone who managed to attend, and that brings us to the end of our Mine Dust and Health Network sessions for this year. Looking forward to more sessions in 2022. Thank you.
Thank you, Cleveland. Bye-bye.
Thanks, Charlene. That was a very good presentation and summary overview. I'm very pleased to see your progress on that. And Cledwin, thank you for indulging my long answers. Bye-bye.
You're welcome, Harold.